Hello, hello. Today we are diving into the structure of the skeletal muscle, one of the most fascinating and adaptable tissues in the human body. Whether you're walking, typing, or lifting weights, skeletal muscle powers every voluntary movement you make. Understanding how skeletal muscles is built help us see how movement happens, why training changes our body, and what happens when injury or fatigue set in. As scientists often say, skeletal muscle is the engine of voluntary movement. But it's not just about strength, it's about coordination, adaptability, and incredible cellular precision. In this lesson, we'll explore how the structure of skeletal muscle supports everything from a simple step to Olympic level performance. Is essential for maintaining movement, posture, and joint stability, and body heat. All, Altogether, the body contains over 600 muscles, each working together to convert chemical energy, also known as ATP, into mechanical energy that allows us to move and function every day. There are three main types of muscle tissue, each with distinct structures and roles. First is skeletal muscle, which is voluntary, striated, and attached to bones by tendons. It's responsible for movements we continuously control, like walking, typing, or lifting. We also have cardiac muscle, which is involuntary. It's also striated and found only in the heart. Its rhythmic contractions keep blood circulating throughout the body. And finally, we have smooth muscle, which is involuntary and non-striated. It's located in the walls of hollow organs such as the stomach, intestines, and blood vessels. It helps move substances throughout the body without conscious effort. Skeletal muscles is especially unique because it's the only type under voluntary control. It contracts when we decide to move, allowing for precise, rapid, and coordinated actions. Organized much like a set of nested Russian dolls, with several layers working together to create movement. Each level plays a critical role in the muscle's ability to contract and generate force. Let's break it down step by step. The muscle itself is the entire organ, such as our biceps or quadriceps. It's wrapped in a protective layer called the epimysium. Within the muscles are fascicles, which are bundles of muscle fibers surrounded by perimysium. Each muscle fiber is actually a single elongated muscle cell. It's cylindrical in shape and capable of contracting when stimulated. Inside each fiber are many microfibers, the thread-like structures that contain the machinery for contraction. These myofibrils are made up of repeated units called sarcomeres, which are the functional units of contraction. Within each sarcomere are myofilaments, such as actin, which is the thin, and myosin, which are thick proteins that slide past each other to create movement. Now you may be saying, why does this matter? This hierarchical organization ensures that the force generated at the molecular level within the sarcomere is transmitted efficiently to move entire limbs. As bundles of fibers, they're carefully wrapped and protected by three layers of connective tissue. These layers provide structure, support, and pathways for communication throughout the muscle. The epimysium surrounds the entire muscle. It's a dense, tough connective tissue layer that protects the muscles as a whole. The perimyosum wraps around each fascicle or bundle of muscle fibers. This layer carries blood vessels and nerves, ensuring each fascicle gets the nutrients and signals it needs. The endomyosum encases individual muscle fibers. It's a thin, delicate layer that contains capillaries and nerve endings, supporting the exchange of oxygen and nutrients directly at the cellular level. Together, these connective tissue layers not only support and protect the muscle, but also merge to form tendons, which are anchor muscles securely to bones. Which anchor muscles securely to bones. Thinking of them as the packaging system that keeps every component organized and functional, while also serving as communication pathways for nutrients, oxygen, and neural signals that keep muscles alive and responsive. Fiber is actually a single muscle cell, long, cylindrical, and multinucleated. Some fibers can reach up to 30 centimeters in length in humans, making them amongst the longest cells in the body. Each muscle fiber are specialized structures designed for efficient contraction and rapid communication. The sarcolemma is the cell's membrane of the muscle fiber. It conducts electrical signals, helping trigger contractions. The sarcoplasma is the cytoplasm of the muscle cell, containing stored glycogen and myoglobin to fuel contraction. The sarcoplasmic reticulum is a specialized network that stores calcium ions. When a muscle contracts, the SR releases calcium, setting the contraction process in motion. T-tubules are deep invaginations of the sarcolemma that carry electrical impulses into the interior of the muscle fiber, ensuring every myofibril receives the signal to contract. Myofibrils fibrils are within each fiber that contain sarcomeres, the actual contractile units responsible for generating force. Why does this all matter? When a signal arrives, T-tubules carries it deep into the fibers, prompting the sarcoplasmic reticulum to release calcium. This calcium activates the myofibrils, causing contraction, all in just milliseconds. And each fiber functions as a self-contained contraction unit, capable of rapid and precise responses. Is the smallest functional unit of muscle contraction. It's where the real work of movement happens. Under a microscope, the sarcomere gives the skeletal and cardiac muscle their striated appearance. Each sarcomere is precisely organized and defined by several key structures. The Z line marks the boundary of each sarcomere and anchors the actin, which is the thin filament. The M line is the center line where the myosin, which is the thick filament, are anchored. The A band represents the length of the thick filaments, including areas where the actin and myosin overlap. The I band contains only actin filaments and is located between adjacent A bands. The H zone is the center of the A band, containing only myosin, with no overlap of actin. Together, these regions form a repeating pattern that allows the sarcomere to shorten and lengthen during contraction and relaxation. Sliding filament theory. This theory explains how muscles contract. When a muscle is stimulated, calcium ions are released from the sarcoplasmic reticulum, and myosin heads bind to actin and pull the filaments inward. This action shortens the sarcomere, creating contraction. The process requires both ADP and calcium to trigger the interaction between actin and myosin with a signal from the brain. The signal travels down a motor neuron until it reaches the neuromuscular junction, the connection point between the nervous system and a muscle fiber. At this point, a sequence of events takes place in just milliseconds. The motor neuron releases a neurotransmitter called acetylcholine into the synaptic cleft, and a tiny gap between the neuron and the muscle fiber, ACH, binds to receptors on the sarcolemma or muscle cell membrane. This binding triggers an electrical signal or action potential which travels along the sarcolemma and deep into the fiber through the T tubules. The sarcoplasmic reticulum then responds by releasing calcium ions into the sarcoplasma. Calcium binds to troponin, a regulatory protein on the actin filament, causing tropomyosin to shift and expose the binding sites on actin. Myosin heads attach to these exposed sites, pulling on the actin filaments, and contraction begins. Connective tissues such as epimyosin, perimyosin, and endomyosin help distribute mechanical stress across the muscle, reducing the risk of injury. Different fiber types play specialized roles, such as type 1, which is slow twitch, are built for endurance and rely on aerobic metabolism. Type 2 fibers are designed for power and speed using anaerobic energy systems. The body's ability to adapt is what makes skeletal muscle so remarkable. Strength training increases the density of myofibrils and the diameter of muscle fibers, leading to greater force productions. Endurance training boosts the number of mitochondria, capillaries, and oxidative enzymes, improving fatigue resistance and oxygen efficiency. Muscles adapt to the demand. It remodels itself based on how we use it. This knowledge helps us understand how to train, how to recover, and how to treat muscle related conditions. In sports medicine, knowing about muscle fiber types allows trainers and coaches to design targeted programs, building endurance for long distance athletes or increasing strength for sprinters and weightlifters. In rehabilitation, knowledge of muscle structures guides physical therapists to develop recovery protocols after injuries and surgeries. Electrical muscle stimulation can be used to activate or rebuild muscles, and they help restore strength and mobility. In clinical treatments and research, conditions like muscular dystrophy and myopathies involve defects in the structural proteins of muscle fibers. Researchers study sarcomere mutations to better understand and treat genetic muscle diseases. Ways is that the skeletal muscle is highly organized and responsive. Its structure is from myofilaments and sarcomeres to fibers, fascicles, and connective tissue. It allows muscles to generate precise, powerful movements. The neuromuscular junction is a critical link between the brain signals and contractions, ensuring that voluntary movements happen seamlessly. Training, therapy, and rehabilitation can actively reshape muscle structures, whether by increasing fiber size, enhancing endurance, or restoring function after injury.